Okay. So I think everybody can see the screen. I will also try. Oh, okay. I will also try to remove this. Okay. Hopefully it should be fine. Okay, so I will start. Yeah, as Dirk said, I'm Michele Partipilo, PhD student at University of Groningen, and well, as the title of the presentation says, I'm gonna discuss about establishing a minimal pathway for the regeneration of Redox cofactors in synthetic cells. Well, to start, I would like to mention that I'm involved in a bigger consortium that is called BaSyC, or Building a Synthetic Cell. Probably most of you already know about it. That is a consortium of Dutch universities that is trying to reach an ambitious goal, indeed, that is the one of, by the use of a bottom-up approach, trying to rationally engineer synthetic cells, assembling molecular building blocks. And well, the idea is that this system eventually will have the same properties of living cells as the capacity of growth, of replicate itself, and of course to divide. Now, being a very ambitious goal, we decided to divide this task in different sub-modules. But here at the University of Groningen, we mostly focus on the construction of a minimal metabolism. This means that we are trying to find the best way or more optimal to fuel cells, not only with energies, but also with building blocks. And already some work has been published in this regard, for instance, on how to provide ATP in synthetic cells or to ensure the osmoregulation of them, or assembling and forming building blocks for the biosynthesis of phospholipid that enclose the compartment. But as I mentioned at the beginning, I will focus on redox power and redox reactions. Now, the reason, and when we are talking about redox reaction, we are mostly talking about specific molecules. These molecules are nicotinamide adenine dinucleotide cofactors as NAD and its reduced form, NADH, but also the molecule called NADP and its reduced form, NADPH, are fundamental for transferring electron from reduced metabolites to oxidized one within the metabolic framework. And if we look, for instance, living cell and their usage of metabolites, this, for example, is a panel coming from a study in Escherichia coli, we can see that only few metabolites are involved in different reactions. And instead, there is a series of special molecules described as hub metabolites that are instead involved in hundreds of different reactions. And between these hub metabolites, NAD plus and NADH are included, as well the version of this cofactor with phosphate. And if we look at what are fundamental processes we know for living system to be indeed alive, we can notice how reducing equivalents are generated in the form of NADH. For instance, when you try to break down a molecule of sugar, or when there is fatty acid oxidation, we obtain reducing power in the form of NADPH. On the other side, this reduced cofactor will be reoxidized for other important processes, as for instance, the respiration, in which NADH provides electron for the respiratory chain, or as well for anabolic reaction. So when cells are trying to build up complex molecules needed for life. So the goal of my main project, that as Dirk was saying, it's almost 3 years that I've been carrying out, is indeed about trying to design a minimal pathway that ensure the reduction, but also the oxidation of this redox cofactor NADH and NADPH in cell-like systems, so in our synthetic cells. So I will start trying to describe the pathway, and you can see in here a schematic about that shows our three enzymes enclosed in the phospholipid vesicles, but the whole system is started by formate provision. Now, formate is an appealing molecule for different reasons. The two main reasons is that first is an ideal electron donor because it has a very negative reduction potential, but also is permeable to phospholipid vesicles. This means that by adding formate to the vesicle that contain our enzymes, we can start our redox reaction. And the inclusion of the first enzyme, formate dehydrogenase, here reported as FDH, allows the initial step of our pathway, that is the reduction of the pool of NAD+. As you can see, NAD+ is reducing to NADH, but what we see for what concerns the formate is first its permeation and then its diffusion out as CO2. This means that eventually the only thing that is retained by the trigger of our pathway are the reducing equivalents under the form of the redox cofactors. This NADH accumulation from the first reaction can then be used by a second enzyme, here reported as STAJ, that is a soluble transhydrogenase. And this enzyme will ensure the reduction of the other biological cofactor NADP+ into NADPH with the contemporaneous reoxidation of our initial pool of NAD+ that becomes again available for the first reaction. Eventually, since NADPH is extremely useful for reductive metabolism, we decided to include another process downstream, in this case, the glutathione reduction, that is catalyzed by the action of our third enzyme, GORA, glutathione reductase, that indeed will catalyze the reduction of glutathione disulfide, here reported as GSSG. But the other effect that we will get from this final step is the reoxidation of the pool of NADP+, that is again available for the second reaction. So our pathway can sustain its action with the three reactions taking place in the lumen. Since we are working with a bottom-up approach, we had then to overproduce these three proteins in bacterial cells, and then we purify them to homogeneity, as you can see from the three size exclusion profile that I included in here. And the last thing that I want to mention is that, of course, we decided and we chose homologs of these three enzymes that were kinetically compatible between each other, but we also took into account in the design of our pathway the choice of our reactants, since electron move from a more negative reduction potential, as in the case of the couple formate CO2, and they flow to a more positive reduction potential, as in the case of our final acceptor, that is glutathione disulfide and reduced glutathione. This standard reduction potential can be then transformed, rearranging these numbers in the Gibbs equation in equilibrium constancies values. And you can see how the first and the third reaction are favored in the direction of the products, so CO2 and reduced glutathione, while instead our second reaction goes mostly to equilibrium. So the take-home messages from our design is the fact that we rely on formate permeation, so we don't need any membrane proteins that allows the uptake of our initial trigger, but also we design a pathway in a way that would have been not only thermodynamically, but also kinetically feasible. So the first step was, of course, trying to understand if we could carry out the reduction of the initial pool of NAD+ into NADH by the action of the formate dehydrogenase FDH. This is why we started to work with large unilamellar vesicles extruded with the 400 nanometer filter, and we encapsulated in these vesicles a fixed amount of formate dehydrogenase and NAD+. What then we tried to test was trying to tune the rate of the luminal NAD+ reduction, adjusting the amount of formate. And you can see how the rate of the reduction of NAD+ will increase with the amount of external formate we provided. What we are monitoring in this panel is relative fluorescence units. And the reason why we're monitoring by fluorescence is that, as you probably know, NAD+ is not fluorescent at all, but it's the product of our reaction, NADH, is instead autofluorescent. So in here, the readout is basically the product NADH. Last thing that I want to show in this panel is that we also managed to use an external inhibitor that is called thiocyanate. In fact, this molecule is a small permeable molecule to phospholipids that is able to not only to permeate inside the vesicles, but then to bind and inhibits the formate dehydrogenase. So you can see the difference between the reaction in absence of inhibitor using 5 millimolar formate, and instead the reaction in the presence of inhibitor that really slows down significantly the reduction of the cofactor. In the other way around, we also try to fix the amount of external formate provided and fixing the amount of protein that was encapsulated. And we try to observe if we could also vary the amount of cofactor that we could reduce. And as you can see, not only the initial reduction rate changes according to how much NAD+ you have inside your vesicles, but also the final amount of fluorescence is proportional to the concentration of the cofactor that you encapsulated. So these experiments demonstrate us that we could carry out the reduction of NAD+ inside our vesicle. But of course, something that we could not exclude by default was that something else could happen outside our vesicles. And the reason why we started to consider this possibility, so external reduction of NAD+, is the fact that there are different reports about stickiness of proteins, but also of substrates when you prepare large unilamellar vesicles by freeze thawing. For this reason, we carried out negative controls in which we were encapsulating or only the cofactor or only the protein formate dehydrogenase. And then, as I'm going to show you now in this experiment, for example, when we encapsulated only the protein and on the outside we added our NAD+, at the moment of the addition of formate, we observed something that we would have preferred not to see, that is indeed reduction of the cofactor. This meant that basically we had still some of the protein that was sticking on the outside. And also when we tried the other configuration, when we had the spatial separation between the encapsulated cofactor and we had external protein, and we added formate, we saw the immediate reduction of the small portion of NAD+ that was still sticking on the outside, that then, of course, keep the its value of fluorescence quite constant because all the fraction was reduced. For this reason, we had to devise a system that would have avoided to observe any product formation on the outside. And we came up with the scavenger system or external scavenger system that is composed of an enzyme called malate dehydrogenase and a membrane impermeable substrate that is oxaloacetate. Now, in the scenario in which we had our formate and we have some protein or some cofactor still sticking on the outside, we will have NADH form on the outside of our vesicles. But because of the action of our scavenger system, MDH and oxaloacetate, what we will have is immediate reoxidation of NADH into NAD+ with the concomitant reduction of oxaloacetate in malate. And when we try to carry out the negative controls that I mentioned before in the presence of the scavenger that corresponds to the blue line, you can see how we indeed stop to have any development of fluorescence that was coming from the outside. So eventually the presence of the scavenger system was preventing any extraluminal reaction. A last thing we also try to assess was that was whether our scavenger system might affect also the luminal reaction, because in here, in these two panels, I'm showing negative controls, but in our normal setup, where we have the protein in the cofactor, and then we add formate, we would have liked to see if our scavenger was, to some extent, modifying the amount of luminal NADH formation. And indeed, it looked not like the case. So eventually, we also obtained the final proof that the luminal reaction was not affected by our scavenger, as eventually the difference you can observe in this panel is not significant. Before to continue to describe the pathway, we also try to consider another aspect that is important when you talk about synthetic cells, that is the size. We started to work, as I mentioned, with the large unilamellar vesicles with a diameter of 400 nanometer. That, of course, give you some advantages as dealing with pretty robust vesicles, but also the fact that the signal you get come from hundreds of thousands of vesicles, so you really have a high throughput analysis. But on the other side, if you scale up the size of your vesicles, you can start to do other things, as for instance, observe directly your vesicles by light microscopy. And you could also carry out then single vesicle analysis. Another consideration, if we think about volume, is the fact that thinking about having a eventual synthetic cell, we need to have more volume to incorporate more functions. So to incorporate all the proteins that we need, all the folded DNA or ribosomal system. That's why eventually we were also interested in observing if we could carry out our reaction in bigger vesicles. For this reason, I started to team up with a postdoc in our lab, Ella, that indeed is able to work with the giant unilamellar vesicles. In here, you can have a short idea of the protocol used for the giant unilamellar vesicles preparation. And when you prepare them, you can also take advantage of another factor. So the fact that you can work with a microfluidics chip. And of course, in here, as you can see from the design of our microfluidic chips, what you can start to do is to trap your vesicles and then to concentrate them in these bucket-like designed traps. And because they are fixed in this trap, you can start to exchange the external solution. That, of course, was something that we were interested. So what I'm going to show you now is a small movie of a classic experiment in which we encapsulated the protein that I told you, formatted hydrogenase and NADPH in these giant unilamellar vesicles. The reason why you can see this red label is the fact that our phospholipids in here are labeled with a fluorescent dye called ATO633. And the vesicles were first trapped, then washed. And after washing, what we started to do was to exchange the external solution with a solution that was containing formate. And what you can see is that over time, we start to observe an increase in fluorescence. I hope you can see from the movie now. You start to see the increase in fluorescence only in the lumen of the vesicles. So even what I mentioned before about the necessity of having a scavenger system is something that you do not need so much, because you can see that there is no reduction taking place on the outside of our vesicles. You can, of course, plot this nice movie in a Cartesian graph following the increase of fluorescence over time. But then we also decided to take advantage of this feature of exchanging external solution in another way. So we decided to carry out a similar experiment in which initially we flow only buffer. So as you can see on the y-axis, there is no increase of fluorescence when you flush only buffer. While instead, in the moment we exchange the external solution with a same solution, just with the supplement of formate, we could start to observe the increase in fluorescence corresponding to this luminal reaction that I mentioned. And of course, what we could do then was to play around and switch with different cycles of solution with the buffer and solution without the buffer. And of course, this give you an idea also of how having bigger vesicles that can be trapped in this microfluidic device can give you an extra level of control in terms of tuning the electron flow inside the lumen of our vesicles. Okay, let's go back to the pathway. So, as I mentioned at the very beginning, of course, NADH and NAD+ are very important for transfer of electrons, but they are not the only factors. We also need to include the NADP+ and NADPH for reaction involved, for instance, in anabolic processes. The problem when you are in a system that includes both NADH and NADPH, though, is that if you look at these molecules by absorbance, for instance, or by emission fluorescence, what you see is no difference at all. So basically, you cannot distinguish between these two molecules. For this reason, we started to work then with a specific fluorescence sensor that is indeed specific for NADPH, with the capacity of changing the excitation spectrum according to the binding of this substrate, while instead is not affected at all by the presence of NADH, NADP+, or NAD+. And as you can see from the panel here on the bottom, in the absence of NADPH, the excitation spectrum of INAP sensor does not show any peak at 420 and instead a peak around 485 nanometer. While instead, when NADPH is present and is bound to the sensor, you will start to see the appearance of a peak around 420 and the disappearance instead of the peak at 485. We then purified this enzyme and started to test if we could observe the reaction of transhydrogenase, but also its coupling to formate hydrogenase. So what you see in here, it's a measurement done in solution, so no vesicles in this case. And first thing, we could observe the transhydrogenase reaction. So basically the transfer of electron from NADH to NADP+. And this condition corresponds to the empty circles you can see on the left panel. We also managed to observe the coupling of the two reactions. So starting from formate, we started first to reduce NADP+, and then this NADH that was formed was oxidized in favor of the reduction of NADPH. And as you can see from the full green circles, the rate of this transhydrogenation, the discouple, is clearly lower than the transhydrogenase alone. We also carried out controls, as for instance, using the thiocyanate that inhibits the formate dehydrogenase. And in fact, what you see in the pink line is a very low rate of NADPH formation. This is because we are blocking the upstream reaction. But also when we tried to check the specificity of the sensor for NADPH, for instance, monitoring the FDH reaction, here shown in black, you can see how no change of the ratiometric signal and then no formation of NADPH could have been detected. So we have the proof that this sensor was ideal to monitor the transhydrogenation inside our vesicles. And indeed, well, here is the result indeed of the coupling that you can see before adding formate, the dashed line, and after adding formate at the time points that corresponds to 40 minutes in the solid line instead, quite similar to what was reported in literature. Now, once we were sure that the sensor could work to monitor transhydrogenation, we equip our large unilamellar vesicles not only with the enzymes of our pathway, but also with the sensor, of course. So in here, you have the condition and the concentration used for this experiment shown in this panel. And as you can see, because of the INAP sensor, we could observe and monitor the formation of NADPH in our LUVs. In here, the full reduction of NADPH was achieved after 20 minutes, while instead our negative control, so vesicles where only NADH formation was taking place, did not show any NADPH formation as we were expecting. Now, the interesting piece of information you can also obtain from these two samples is also hidden on the other part of the spectrum. So if instead of focusing on the wavelength that are of interest for the sensor, but you go to look at the wavelength of the reduced cofactor as 370 nanometer, for instance, you can still observe the cofactor reduction that is mediated by formatted dehydrogenase. In this case, this is a 1 millimolar NADH formation, as you can see, when the reaction reached the steady state. And we could observe the quantitative difference with the coupling, where instead we had the reduction of 1 millimolar NAD plus and 0.2 millimolar NADP for a total pool of 1.2 millimolar cofactor that are reduced. So we can still retain information on the reduced cofactor, although on the right graph, we do not know what is what, while on the left panel, we know that we are detecting clearly NADPH. Also, in this case, we scale up the size of our compartments. So we carried out a similar encapsulation to what you have in here, but in giant unilamellar vesicles. And what I'm showing now is not anymore a single wavelength, but I'm going to show you what happens when, after dropping the vesicles, you flow formate and you follow the ratio of 405 nanometer and 488 nanometer. So in this case, there was no overlay, and I cannot show you the membranes, but I hope that you could see what is shown instead in here in the Cartesian graph, where the presence of NADP plus can be detected using the ratio 405 for 88, while instead when we we tested a condition where everything was included, the two enzymes and NAD plus, but not NADP, you have only a negligible increase in the ratio, mostly coming from the small increase at 405, because we are still reducing this NAD plus into NADPH. Well, eventually, as I told you at the very beginning, we do not want only to reduce cofactor, but we also want to find a way to reoxidize them. And that's why we chose a final drain. Our drain is, as I mentioned, disulfide glutathione reduced into the reduced glutathione indeed. So we first tested if we could observe this process, and we monitor, in this case, not the reduced cofactor or the oxidized cofactor, but we monitored the glutathione reduction using an Hellman assay, here reported as DTMB. So if we compare the activity of glutathione reductase alone, so NADPH that transfer electron to glutathione disulfide, you can see from the panel present in presentation that corresponds to the empty circle that the reaction is extremely fast, as you could expect. While instead, when we couple our oxidized cofactor, NAD plus and NADP plus, and our three enzymes, and then we triggered the pathway action by formate addition, you could see a much slower rate of glutathione formation that still was taking place as expected. And in this case, we show how changing the amount of one of the three enzymes, in this case, the transhydrogenase, we could observe a different reaction rate. So according to the amount of transhydrogenase we use, we had a full reduction of our glutathione pool that was going from 15 minutes with 0.2 micromolar STJ to 1 hour using 5 times less enzymes. Also in this case, we had our negative controls, again, the thiocyanate in pink that inhibits the first enzyme, the formatted dehydrogenase. We also carried out experiment in which we used the full pathway, so all these enzymes, but devoid of one of the two cofactors. And you can see that when we did not include any NAD plus in this case, the empty triangle, we fully abolish any glutathione reduction at all. While instead, when we did not include only NADP+, we had, although at a very low rate, glutathione reduction. And the reason because we observed this behavior is that the glutathione reductase enzyme, although specific for NADPH, can also accept some electron from NADH with, of course, a lower specificity and a lower catalytic efficiency. Well, although it's nice to monitor the final product, we add our sensor, our NADPH sensor that we were also using in vesicles. So we also try to monitor it in solution again, not the final product formation, but the intermediate dynamics. And as you can see from the panel on the right, we could observe a difference increasing the amount of our glutathione disulfide, our drain. So you can see how in the absence of any drain, the reduction of NADPH was taking place extremely fast. I think here was around 40 minutes, while instead you can see the effect of including a final acceptor. So for instance, at 0.1 millimolar glutathione, you start to see this first steady state of NADPH that corresponds to a very low amount of NADPH because all the electrons are accepted by glutathione disulfide. Once the full pool is fully reduced to reduced glutathione, formate can finally provide electron to NADPH that correspond to this very quick increase in fluorescence. And then it reached the final steady state. And you can see how the more we increase the drain in terms of glutathione concentration, the more we observe a delay in our steady state phase, till reaching a point in which we had an excess of glutathione that just could not match any NADPH formation at all. And this explains, for instance, what you see in the condition of 2.5 or 5 millimolar disulfide glutathione, for which even after 5 hours, we could not observe any NADPH form because all the electrons are going to the final drain, that is the reduced glutathione. Finally, we equip our large unilamellar vesicles with the full pathway, and also in this case, our setup was trying to observe a difference increasing the final drain. As you can see, in the condition in which we included only the 2 first enzymes, we had a very quick reaction, so a very quick NADP+ reduction, while instead, when we started to include the drain for this electron, again, glutathione disulfide, you can start to see a delay of this NADPH reduction that, of course, increase with the amount of the drain till reaching what I described already before as a long steady state phase of the NADPH. And a similar experiment was carried out also in giant unilamellar vesicles, although in this case, I just reported 2 extremes of no drain at all and a big drain that just prolong the steady state phase of an NADPH generation, since all the electrons are indeed going into the direction of the final acceptor. A last thing I would like to discuss today, in terms of experiments at least, is how stable is our pathway once it is incorporated in large unilamellar vesicles. And the reason why we wonder that is that, of course, in this case, we are only testing enzymes with reactants. We do not have any system to renew the pool of our enzymes. So how long can we expect the enzymes to be effective in the compartment of large unilamellar vesicles? For this reason, we measure the activity after the first day of preparation of the vesicles, and then we store them at 4 degrees. And the day after, we try to carry out the same experiment in which we were again triggering the NADPH formation. Although, of course, we are mostly talking about the full pathway activity. As you can see, the activity of our vesicles was retained almost completely after 3 days of storage at 4 degrees, but still after 7 days, they retained quite remarkable 60% of the original activity. After 2 weeks, we started instead to observe a significant drop in the activity of the vesicles that eventually just did not manage to give us any kind of change in the ratiometric signal after 3 days. We try to understand if this kind of behavior was something that we should have ascribed to the stability of the vesicles, or if it was a phenomenon that was regarding the enzymatic deactivation. For this reason, my colleague Jacopo performed dynamic light scattering on these vesicles, and basically he managed to observe that the distribution profile of the vesicle at the day of the preparation just did not change after 21 days of storage, as you can see here. So we assume that most of our vesicles just maintain their structural integrity, reason that make us think that then it was something that was going on with the enzymes that instead were incorporated. For this reason, we started to look not anymore at the hydrodynamic radius, but also to the excitation spectrum of our vesicles. And you can see that the excitation spectrum we were obtaining was mostly conserved, identical after 7 days. But then we started to observe a peak appearing around 400 nanometer. A peak appearing after 14 days, a peak that became more prominent after 21 days of storage. For this reason, we took the approach of taking our enzymes and test them in the same condition of the vesicle. So we stored them at 4 degrees, and we then observe if one of the single enzymes was changing the optical properties. And indeed, we found out that there was an enzyme that was behaving in a similar way to what we observed from the vesicles incorporating the full pathway. And this enzyme was the soluble transhydrogenase. This enzyme, indeed, after 11 days of storage, did not change at all the absorbance spectrum, but after 20 days of storage, started to show a similar peak to what we observed in the vesicles incorporating the full pathway. So eventually we figured out that what was happening is that the transhydrogenase was aggregating over time, changing the optical efflorescent properties and losing activity. So eventually it's the transhydrogenase that limits the activity of our redox regeneration pathway over time. So I finally reached the conclusion of this talk, and I hope at this point you agree with me that we managed to develop an enzymatic pathway that is able to ensure the reduction but also the reoxidation of nicotinamide cofactors. This pathway works in solution and in cell-like system regardless of their size, so both in LUVs and GUVs. Very curious is the fact that one year ago, almost 2 years ago, we encountered a paper in which they also were using in vivo, though, the combination of a formated hydrogenase with a transhydrogenase. And this system was giving bacteria a new property, that is the capacity of growing on formate. So while instead formate at high concentration is toxic for living system. So this is quite appealing in terms of applicability of our pathway, maybe also for in vivo systems. Other consideration and conclusion is that our pathway is definitely based on formate permeation, and we need only a small amount of formate to trigger the activity of the pathway at an optimal rate. So this is quite good if we think about possible osmotic stresses if we instead would need a higher amount of formate. And indeed, we also are able to monitor in vitro the activity of the pathway, mostly as NADPH dynamics, although I show you that you can also observe activity as reduced cofactors, but the cool thing of monitoring specifically NADPH is that you can really observe the effect of a drain on a system, on our pathway. And of course, thinking about the integration with the more complex systems, more complex modules, you could think about coupling these processes to NADPH and to observe their activity in function of NADPH. For the future, what we are already starting to do, but we plan to do, is to integrate our redox pathway with other metabolic modules in a way that we can obtain a functional and minimal synthetic metabolism. Since our pathway is formed of only of 3 enzymes and is triggered by the main event of formate oxidation, of formate permeation, also our pathway might be ideal for modeling, since the small amount of enzymes might help for not only for understanding properties that maybe we did not analyze yet of our pathway, but also thinking about integrated in an optimal way with other metabolic functions. And the last thing I would like to mention for the outlooks is that in living system, NADH and NAD+, as well, NADP plus and NADPH, are used to adapt the regulation of translation and transcription in for adapting the cell cycle or to adapt to different environmental stresses. So eventually we could consider to use this pathway activity to regulate the transcription translation responses in our final artificial cells. And of course, the acknowledgements for this work. I would like naturally to thank my supervisors. So first of all, Dirk, that introduced me also before, Professor Dirk-Jan Slotboom, but also Professor Bert Pullman, that contributed as well. A big thank also to Ella, since she made all the work in giant unilamellar vesicles, but also a few nice graphs that you saw today were done by her. The help of Jacopo with the dynamic light scattering measurements, and the consulence of Professor Marco Frey in understanding that aggregation was the limiting factor of our pathway. And here you can see the last picture 2 years ago of our group here at University of Groningen called Membrane Enzymology. And I would like to thank even slightly more all the people that are involved in the subgroup of the SynCell meetings. And last, I would like to thank the people from the BaSyC consortium, since they made possible having this nice story here today.
Thank you very much, Michele. It's a real pleasure to see the complete story, because we've been involved with this work for so long that sometimes you only see the little details, but to see it as a complete story presented by you in a delightful manner, that's really great. Now there's a lot of questions in the chat here, so I suggest that I will read them out and then ask Michele to try and answer them. But you can also raise your hand, and then I will try to to switch between the chat and the raised hands if you just want to speak using your microphone. So I just start on the top, and on the top there's a question on lamellarity and size. So there's a question on whether it would work in smaller vesicles. You've shown bigger ones, but whether it would also work in smaller vesicles, and what would happen if they are multilamellar.
So first of all, I would like to say that although for instance, our large unilamellar vesicles are mostly unilamellar, you always have a small percentage of multilamellarity. So I would say that should not matter to a significant extent, but of course it would depend on how this multilamellarity affect also the encapsulation of the enzymes and of the reactants. In terms of size, could you do this in either smaller vesicles? I think you could. Well, we demonstrated that we can work from a few hundred nanometer diameter up to tens of micron. So I don't see a reason why it should not work with smaller vesicles, although then you would have the problem of having a point for which your volume just cannot allow the incorporation of all the reactants and enzymes that you want. And I guess that they might work in multilamellar system, but it depends to what extent they are multilamellar, I would guess.
All right, so then I will just go to the next question from Anders Bennett. Fascinating talk. What is the dependence on type of lipids in the vesicles? So what lipid compositions might you be able to use in this system?
So I think somewhere I have the lipid composition, but I can tell you that my colleague Jacopo, yes, so here we have the composition of the composition of the LUVs. We mostly work with the DOP kind of lipids, and I think that the work that was published by Matteo Gabba and Jacopo Fraricciardi shows that you can play around with different lipids, particularly for formate, because formate is quite fast in permeating synthetic phospholipids. So I think that it really depends on what molecule are you using to trigger your system. Formate should be quite flexible, I think, for different lipid composition. But of course, you might slow down the permeation that usually is quite fast, depending on how long are your lipids, the level of saturation in saturation, and so on.
Yeah, and it will probably become more of an issue if you have membrane proteins reconstituted in your system as well, which you of course have, which is great, but in future systems we probably will need. Then there's a next question from Bineet Sharma. Nice work. So you get a lot of compliments, Michele. So there's a question on why the GOVs stick to each other in the wells in your experiment, and then again, what's the lipid composition? But that you just answered. So why they stick to the GOVs? So
the lipid composition is very similar. Just there is a very small percentage, I think less than 1% of these label phospholipids for see them, to observe them. The reason why they stick is that they are trapped. So basically imagine that you have different traps in which you have your vesicles, but you have a constant flow of, well, could be KPI with the formate. So they are basically just trapped. Of course, there is a small extent in which you can see that they fluctuate slightly, but mostly they are just in contact with each other because they are trapped and there is a flow applied. So yeah, if I re-show the video, you can see that they just slightly move, but mostly they are stiff because of that.
Okay, then I move to the next one. There we have Brendan Mortage. Would the system also work with coacervates or other membraneless assemblies?
Membraneless assemblies. Well, I think it depends on the level of permeability of these assemblies. But I think that for instance, I can think about polymerosome, so where you have a mix of lipids and polymers. I think formate permeates also in that case, although to a slower rate. But yeah, I don't know if you refer specifically, you have some specific case in mind, because maybe that could help to answer the question.
Now we'll have to see whether Brendan will quickly answer your counter question or not. I'll give Brendan 2 more seconds.
But I would expect that might work. I would expect that, unless there are main physical reason why formate cannot go through.
Yeah. Of course, you haven't tried, but it's something that a priori there's no reason to think why it would not work.
True.
Then let's move to the next question from Emma Jones. This is an awesome talk. Again, a big compliment, Michele. Would it be possible to integrate that pathway with protein expression somehow, or express the enzymes from more complex synthetic cell genome?
Well, this is what I hope. Like this is, to be honest, one of the next steps and something that I hope we will manage to accomplish in a short amount of time. But the ideal scenario would be having a small genome that is able to codify for this protein. So you produce the protein within the system. But sadly, I did not manage to work with any DNA encapsulated so far. And but yeah, that would be the ideal application, to be honest.
Yeah, of course, that is something that that this work will hopefully lead towards. But of course, that increases the complexity tremendously. But hopefully sooner rather than later.
Yeah.
Yeah. So then there's a question from Peter about the pH dependence of the pathway. So what are the boundaries in terms of pH that you can use this pathway?
So for the full pathway, I think with a good level of confidence, I can say between pH 6 and pH 8, because now I'm thinking about the pH optimum of the three enzymes that we use. So the formatted hydrogenase, the transhydrogenase, and the glutathione reductase. In terms of lipids, I also don't think they would be strongly affected by this kind of pH, but if you go, for example, too low in pH, I would expect that the transhydrogenase starts to lose activity. While instead I can tell you the formatted hydrogenase is extremely active in a very broad range of pH. So I would say between pH 6 and pH 8, I would strongly think that the pathway is active to in an ideal way. After this range, I should personally test and tell you.
All right, then we move to the next question, and that is about the scavenger system. Would it be possible to treat deformed LUVs with a protease rather than using the scavenger system to get rid of the external FDH at least?
Yes, so this was another idea. I remember also another professor mentioning this, and this is a great idea. So the answer is yes, but a limiting factor in there is that what I like also of the scavenger system is that it's very specific for the problem. So you have a dehydrogenase that is basically able to get rid of the reduced NADH, reoxidizing it. But if you think about having more proteins, well, imagine a membrane protein also incorporated because you have your synthetic cell with different function. The protease could also be a problem in that case. So I would say for this pathway, yeah, a protease would work fine. But if you have more complex system, the protease might be a problem for protein that are embedded in the membrane, for instance.
Yes. And then there's a question from Patrick, whether you have considered doing these experiments on sort of a scaffold to bring the enzymes together to increase the, well, let's say maybe the kinetics of the consecutive reactions.
I did not try. No, this is something that I did not find time to try, but I think it might be a good strategy to increase the efficiency of some of the reaction. Might be something worth it to try indeed.
Yeah. Then there's a question from Bernard, who really liked the talk as well and said it's a fascinating talk. Actually, Patrick also said it's an excellent project. Congratulations. Could you use this platform to do artificial evolution of those enzymes to optimize all three as a pathway?
Oh, that's a great question. Oh, so this specific platform, like in vesicles, well, I would like to talk privately then about it, because we never use this, we never considered that, but might be something to look into maybe.
Yeah, it is something that we, well, probably occasionally quickly mentioned in the background of trying to optimize this in a cellular system to have these different enzymes together and try and optimize it, but it never materialized so far. But it's a very interesting suggestion. Then there's a question about the rest is all comments on what you've done. So I think we've reached the last real question.
I think there was the counter answer about the coacervate question. I think I can see, I think of membraneless organelles like the intrinsically disordered proteins that self-assemble to coacervate. I should think about this, but till the point the intrinsically disordered proteins are not a barrier for formate permeation, I think it might work, the pathway in such a system.
Yeah, that was a clarification from Brandon on the membraneless organelles. I think it's something certainly worth considering, and also when building more complex systems, it would be good to also consider other options for the enclosed environment. All right, so I don't see any further questions. So I think it was a fantastic presentation, Michele. There was a very lively discussion. So I really thank all the audience as well, because it's great to have all these suggestions and to see how many people are actually interested in this type of work. Sometimes when you're alone in the lab, you may think the world doesn't care, but at least the 50 people who were here today, they do care. So that is absolutely great to see. So thanks all for joining, and hopefully see you in one of the future lectures again. All right, bye-bye.
Bye.
Thank you so much. Bye.
You're welcome. Thanks again for the opportunity. Bye.
Bye.